Next speaker is Tim Erickson, and he will talk about the assembly of the MET complex in sensory hair cells.
Hi everyone, good morning. My name is Tim Erickson. I'm a postdoc in the Nicholson lab at OHSU in Portland, Oregon. And today I'm going to be telling you about a gene named Tomt and how it regulates the trafficking of TMC proteins to the hair bundle in sensory hair cells. So hair cells are the receptors for auditory and vestibular information in vertebrates. And no matter whether you're a fish or a human or what type of hair cell you are, the basics of hair cell mechanotransduction are the same. When a stimulus deflects the hair bundle, this leads to the opening of mechanically gated channels located at the tips of the stereocilia. And in this way, mechanical force is converted into an electrical signal. Now currently, this very small part of the hair cell is the focus of a lot of research to try and understand the molecular and genetic bases for forms of human deafness. Our current understanding is that the MET complex is composed of these proteins, the tippling protein, protocadherin 15, as well as the multi-span transmembrane domain proteins, TIMI, LHFPL5, and the TMCs. Now the big question in the field is what is the identity of the pore-forming subunit of the mechanotransduction channel? And although this matter is not yet settled, the TMCs appear to be the most promising candidates for this. In mice, TMC 1-2 double knockouts have no mechanotransduction, and single amino acid changes within TMC1 can actually change the properties of the MET channel. So for this reason, it's very important to understand the role of TMCs in mechanotransduction and how they form a functional complex with their partners. The zebrafish hearing imbalance mutants were first named for things that orbited. Because these fish have vestibular dysfunction, they tend to swim in circles. Over time, the identity of these mutants was found out, the genes were cloned, and many of them were members of the MET complex. However, there was one mutant named mercury that eluded identification for over 15 years. In a sense, this was like our cloche mutant. It was the unclonable gene with like a perfect mutant phenotype. And to make a long story short, about 2 years ago, we finally cloned mercury and found that it was transmembrane O-methyltransferase, or Tomt for short. The Tomt protein has a single transmembrane domain at its N-terminus, followed by a SAM-dependent methyltransferase domain. We have two alleles of mercury. Both are stop codons. Both cause early truncation of the protein and are predicted to be nulls. Tomt is expressed specifically in hair cells of the inner ear and lateral line. And surprisingly, Tomt protein is not localized to the hair bundle where mechanotransduction happens, but rather is enriched in the Golgi. We have GFP and HA tagged versions of Tomt, and they both localize to the same apical membrane compartment. And we confirmed that this was Golgi by co-localization with a medial Golgi marker. So what else is known about Tomt? Interestingly, it's a known mouse and human deafness gene. It's responsible for DFNB63, which is an autosomal recessive, non-syndromic form of human deafness. And it's a very severe form too. Hearing loss is usually profound and present at birth. The Tomt enzymatic domain is related to a well-characterized enzyme called catechol O-methyltransferase, or Comt. Comt is known to be involved in the degradation of catecholamines, such as dopamine, epinephrine, and it does so by attaching a methyl group to the hydroxyl group and sending it down the degradation pathway. In 2008, there were two papers that described the human and mouse mutations, and their findings are summarized here. Tomt is required for auditory and vestibular function. And the major observation was that the hair cells in the cochlea degenerate and that this presumably causes deafness. Now it should be pointed out that this hair cell degeneration is a common phenotype amongst mechanotransduction mutants in mice. But nevertheless, the authors speculate that it is excess dopamine or some other catecholamine that's damaging the hair cells, causing the degeneration and the deafness. Now, by way of contrast, I'll give you our conclusions from the mercury mutant first and then show you data after. Tomt is required for auditory and vestibular function, but it's not catecholamine metabolism or hair cell degeneration that's causing the deafness, but rather Tomt-deficient hair cells lack mechanotransduction. And we think at the root of this is that Tomt is regulating the assembly of the MET complex by facilitating TMC protein localization to the hair bundle. So to show that Tomt is required for auditory function, we assayed for the auditory evoked behavior response. In wild-type larvae, they exhibit a very robust startle reflex in response to loud sounds. So to quantify this, we followed this stimulation protocol and let a computer track the movements of the fish in response to sound. Shown here are representative traces from siblings and mercury mutants, and the group data is shown here on the right. On average, wild-type larvae will respond 75% of the time to this stimulation, whereas mercury mutants respond approximately 2% of the time. So mercury mutants are deaf. Next question we asked was, can we rescue this phenotype with our Tomt GFP transgene, the protein which is enriched in the Golgi? And the answer is yes, we can fully restore hearing to the mercury mutants with the Tomt GFP transgene. So we were interested, why are mercury mutants deaf in the first place? We weren't seeing any evidence for hair cell degeneration causing deafness. So in collaboration with Jennifer Old, Catherine Hardy, and Walter Marcotti at the University of Sheffield, they performed some recordings from lateral line hair cells and to measure the MET current in mercury mutants. To do this, to assay for the presence of mechanotransduction, they measured the contribution of the MET current to the resting membrane potential of the cell. To do this experiment, they first patch onto a hair cell, and then they measure the resting membrane potential. Then they add a MET channel blocker, DHS. Since the MET current has a depolarizing influence on the resting potential, if you block the MET current, you should hyperpolarize the cell. And if there's no MET current, then the DHS should have no effect on the resting membrane potential. And that's exactly what we see here in the data. Wild-type and mercury mutant hair cells have similar resting potentials, but the addition of DHS hyperpolarizes wild-type cells and has no effect on mercury mutants. So this suggests that mercury mutants have a specific defect in mechanotransduction. We can also assay for MET using FM dyes. In wild-type cells, FM dyes can rapidly enter hair cells through functioning mechanotransduction channels. Once in the cell, they fluoresce brightly and label the cell. In MET mutants, this does not occur, and there's no label of the hair cells. We can take live fish, bathe them in a solution of FM dye, and light up all the functioning hair cells of the lateral line, and then do live imaging to determine whether a particular fish has active mechanotransduction in the hair cells or not. Consistent with the EPHIS data, mercury mutants do not label with FM dyes. And as with the behavioral phenotype, we can fully rescue it with the Tomt GFP transgene. Now, if the defect in mechanotransduction is due to a failure to metabolize catecholamine, such as what was speculated for the mouse and human, you should be able to rescue this phenotype using the bona fide catechol-O-methyltransferase by expressing Comt in hair cells. We did this experiment and found that Comt has absolutely no effect on mechanotransduction. So this suggests two things. First of all, that catecholamine metabolism is unlikely to be the cause for the mechanotransduction defect in mercury mutants. And secondly, that Comt and Tomt might actually have different substrates. So what is Tomt doing then? Because mercury mutants have a specific defect in mechanotransduction, and the Tomt protein itself is in the Golgi and not in the hair bundle, we hypothesized that it might be regulating formation of the MET complex somehow, maybe by regulating trafficking of some protein. We have tagged transgenes for each of these proteins, for protocadherin 15, for TIMI, LHFPL5, and a pair of the TMCs. In wild-type larvae, these transgenes can localize to the hair bundle and can rescue their respective mutants, so they're functional transgenes. So we used these to look in mercury mutants to see if there was a problem. For the next couple slides, I'll be showing you views of the lateral crista, which is a sensory patch in the inner ear of the zebrafish, with their nice tall hair bundles up here, and this box is showing the region of interest here. For all three of these transgenes, protocadherin 15, LHFPL5, and TIMI, they can still localize to the hair bundle in mercury mutants. And the data is quantified here. We do see a modest reduction for a pair of the transgenes, but we think this is unlikely to account for the complete lack of mechanotransduction in these hair cells. We also have tagged versions of TMC2B and TMC1. And you can see here, as indicated by the yellow brackets, that these proteins can localize to the hair bundle. Strikingly, though, in mercury mutants, this GFP signal is completely gone from the bundles. We still see it in the cell bodies, but it's excluded from the bundle region. And this graph is showing the quantification of the GFP intensity in the bundle region only. Now, we were worried that maybe this defect in TMC localization was a secondary effect of a mechanotransduction problem, that maybe all mechanotransduction mutants have this same problem. So we looked in protocadherin 15 mutants, mutants for the tippling protein, and we found that there was no statistical difference in TMC localization in these mutants. So this suggests that there's a specific and unique role that Tomt is playing in regulating the TMCs to the bundle. Lastly, we asked the question, can Tomt rescue TMC localization in the hair bundle? To do this, we mosaically expressed Tomt P2A NLSM cherry in mercury mutants that have the TMC2B transgene. And then at day 4, we asked the question, do Tomt-positive cells have TMCs in the bundle? And so what I'm showing here is the same wild-type neuromast, upper level where the bundles are and the lower cellular level. You can see TMC2B GFP in the bundles of the wild-type cell. In the mercury mutant, there's only 2 cells that have the GFP signal, and these are the 2 cells that are expressing Tomt. So we can rescue the TMC localization, and this lends support to the idea that Tomt is regulating TMCs to the bundle. So in summary, in wild-type cells, Tomt is in the Golgi, where it's facilitating the transport of TMCs into the bundle, where it can form functional mechanotransduction complexes with its partners. In the mutant, there's no Tomt protein. The transport of TMCs does not occur, and functional mechanotransduction complexes do not form. We think that this, rather than catecholamine metabolism or hair cell degeneration, is the actual cause of deafness in the human DFNB63 patients. Of course, a couple of questions remain. Is Tomt acting directly on the TMCs or via another protein? What is its substrate? What is it methylating? We're not sure right now. Hopefully we'll figure these things out. So I'd like to thank the Nicholson Lab, especially Teresa, of course. Italia is presenting a poster on the role of TIMI in mechanotransduction, so go see that. I'd like to thank Rayo and Rachel for making some of the transgenics, and Elizabeth for her initial efforts in cloning mercury. And I'd also like to thank Alex for his role in isolating one of the alleles of mercury, and the Marcotti Lab for the EFIS data. So thanks. I'd be happy to answer any questions.
Is there a decrease in the number of hair cells in the optic hair cells or lateral line hair cells in Tomt mutants?
Yes, it's typical that we'll see a slight decrease in the number of hair cells. Yeah. All right, thanks.